Yeah. Okay, once again, sorry that the videos just don't have the resolution to see what I put on the blackboard. It's, file size versus resolution. It's always a tough one. These files are huge. What was it? Tuesday's lectures, even compressed, were 1.5 gigabytes. That's a lot of data. Yeah, video files are always gigantic. Okay, well anyway, so we're going to take another look, once again, at a few more aspects of facilitated diffusion. We mentioned the basics about how that works. We mentioned that these channels are regulated. You can have voltage-gated channels, meaning changes in the membrane voltage will distort the protein opening or closing the channel. You can have ligand-gated facilitated diffusion, meaning some molecule non-covalently can bind to the channel protein and cause it to open or close. Neurotransmitter receptors are a good example of that. And then finally, you can do it the old-fashioned way. You can regulate them simply by phosphorylating the protein at key points. That can do things like open or close the channel. But let's get a few examples of what these things do. Facilitated diffusion can be used to carry in things like nutrients from the outside, all kinds of different small molecules, and not just from the outside of the cell to the interior of the cell, but also within cellular compartments or organelles. For instance, think of this. The mitochondria in our cells produce 95% of the ATP we need. That wouldn't do us a whole heck of a lot of good if that ATP could not be transported out of the mitochondria. You got high concentrations of ATP inside the mitochondria, and you got lower concentrations outside. If you didn't have ATP transport channels, the stuff would stay in the mitochondria. It's great for the mitochondria. It's really bad for all the rest of you. So we have mechanisms to transport ATP out of the mitochondria through not just one, but through two membranes. And likewise, we have to take ADP in so we can convert it into ATP. So we can do membrane transport across any membranes, including organelle membranes. Okay, well, that's one kind of thing. Ions are another big thing. Cells need to keep ions at certain levels. Think what happens in your own body if your potassium or sodium levels go too high or too low, you end up with a cardiac arrest. Cells keep calcium ions at low levels compared to the outside, and so on. So we have all kinds of transport mechanisms to get ions into and out of the cell. Especially important are things like the ligand-gated ion channels and the voltage-gated ones. These are very important things like nerve transmission, especially ions like sodium ions, potassium ions, and chloride ions. Those are very important in all kinds of physiological processes. Nerve transmission, muscle contraction, kidney function, xylem and phloem if you happen to be a plant. So those are also important things that can get through by facilitated diffusion.